Hey guys, welcome back to another video. I know it's been a while, but I'm back, and today we are going to be talking about the major scientists who have contributed in the discovery of DNA. So the first scientist we're going to talk about, his name was Griffith, okay? So Griffith, he was like some British guy, and he wanted to find out what macromolecule was where the information that was causing pneumonia in mice was. What macromolecule is it? Like, where is the information that's causing pneumonia being stored in these poor mice? So he discovered that there were two different types of pneumonia causing bacteria. There was R strain and S strain. R strain, it was just the bacteria, whereas S strain, it had a smooth capsule around it, thus S for smooth, right? So he was confused on what was making these mice die. Where's the information being stored? Well, he carried out a series of experiments. His step one. So he noticed that there were two different types of bacteria. So what he did is he took test tubes, or like mixtures of live R strain and S strain. So he put live R strain, he injected it into the mice, but he noticed that the mice was still alive once he injected the R strain. So that, it's not the R strain that's killing the mice, that's giving pneumonia to the mice. So, and then he did the S strain, and he noticed that when he injected it into the mice, the S strain, the mice were dying with the S strain. And this is a term called virulent, which basically just means like it causes a virus, because pneumonia is a virus, and then that was what was killing the bacteria. So then he did a second step where he had placed live R strain, and then he had heated up the S strain, which means that it killed off the S strain. So the S strain was dead, and he made a combination of these, and he injected it into the mice. But okay, so the S strain is dead, so why would it even have an effect? But no, still, the mice were still dying. So he came up to a conclusion. He wasn't able to come up with like a proper reason or conclusion, but he did create a process known as transformation. Transformation meant that there has to be some type of factor that causes the disease that was given from the S strain, alive or dead, and giving it to the alive R strain and transforming the R strain into deadly S strain. And then that's when the mice died. So basically it's kind of just like, think of like an infection or like a zombie apocalypse where it was basically infecting it. No matter if the S strain was alive or dead, he noticed that there was some type of factor, some type of macromolecule, some type of cause that's causing the R strain to become deadly virulent S strain. So, and then our next series of scientists that we're going to go over are scientists called Avery, MacLeod, and McCarthy. So they used Griffith's experiment, and they were like, okay, let's figure out this transforming factor. Let's figure out the transformation factor. So what they did, they got 5 test tubes, and they had put all in each test tube heated S strain. Notice how the S strain, it's dead, meaning that the bacteria, the S strain bacteria, is dead because it's heated. And then in each test tube, alongside the heated S strain, he placed a specific enzyme that destroyed a specific macromolecule in the S strain. Like for this test tube, he placed an enzyme that killed off all of the DNA macromolecules in this S strain. He did the same thing for carbs, where he got an enzyme that killed off all the carbohydrates in the heated S strain. Same thing for lipids, proteins, and RNA. And so then he said that, okay, if I inject all of this into mice, and then let me see which ones die, which one alive, right? So basically, and then what they did is that in each of these individual test tubes, he added live R strain. He added live R strain because they're trying to figure out what's the transformation factor, what is the macromolecule, because these are testing individual macromolecules. So they're trying to figure out what's the macromolecule that is transforming the R strain into the deadly S strain, right? So they placed the R strain in each of the test tubes, and then they inject it all into separate mice, not one mice, but 5 different mice. So this one was going to one mice, this was going to another. And then you want to know what they found out? Is that every single mouse died except this test tube, the one that killed off the DNA, which proves that DNA is a transformation factor, because when DNA is not present, the mouse was living. When they put this into the mouse, it lived. So Avery, McLeod, and McCarthy, they figured out that the transformation factor was DNA. The next two scientists, their names are Hershey and Chase. They're a husband and wife team. And so they tested bacteriophage. What are bacteriophage? Well, they are viruses that only attach and infect bacteria. Okay, bacteriophage. So they wanted to know what they wanted to test is what was being transferred to a host cell when a bacteriophage latches onto it, or when it's infected with the bacteriophage. So they observed the structure of bacteriophage, where on the outside, it had a protein coat, and on the inside, it had these structures where it was their DNA was stored for the bacteriophage. And so they noticed that in the protein coat, they had a unique element, sulfur. But in the DNA, it didn't have sulfur, and it had phosphorus as its unique specific element, and vice versa. So those were their unique elements. So what they did is that, they used radioactive elements of phosphorus and sulfur, and they traced the macromolecules of the radioactive sulfur and phosphorus to see what is being transferred. Is it the proteins that are being transferred, or the DNA that's being transferred? And you'll see that in the outcome. So what they did is that they're we need to separate the virus, the bacteriophage, from the cell. So they put it in basically a contraption called a centrifuge, where basically it's a box, and then you place stuff. It's not like a box, and then it just spins it around really, really, really fast, so it separates the two things. And then they noticed that they found traces of phosphorus in the host cells. When they had separated the virus from the cells, they found traces of phosphorus, of the radioactive phosphorus. But which one of these had the radioactive phosphorus? DNA. So they concluded that DNA from virus, that it infected the host DNA, it noticed that the DNA from this bacteriophage was infecting into the DNA of the host cell, and then that was causing the host cell to duplicate and make more mini viruses, and then after a long time, the host cell would kind of burst and just release a bunch of viruses, and then grab infection, things like that. So the fourth scientist, his name is Chargaff. So Chargaff, he's this Austrian guy, and he noticed that in nature, every single living thing, all their DNAs only had 4 different types of nitrogen spaces. And he noticed that every time the amount of adenine always equaled the amount of thymine, and that the amount of cytosine always equaled the amount of guanine. So he said that, so he came up with the base pair rule, which states that adenine is always chemically bonded to thymine in DNA, and that cytosine is always chemically bonded to guanine, meaning the nitrogenous bases, these types of nitrogenous bases are always chemically bonded. The next pair of scientists, their names are Rosalind Franklin, who's a girl, and Wilkins. So, little fun fact, a little tea is that so Wilkins, he created the atomic bomb, which killed millions, and he fell into a really period of dark depression. So he decided that he would discover something that would contribute to life, like DNA. So him and Franklin, they didn't work together, but they worked in the same area at the same time. And they both worked on X-ray diffraction. X-ray diffraction, well, what is it? It's basically how they would take something small, in this case, they took DNA, and they took they put it through an X-ray and got a microscopic image of what they had diffracted, what they have taken a picture of, basically. So when they took a picture of DNA, this is what they noticed. Well, they noticed that it was kind of like the DNA strands were kind of looking like this. It was just kind of like squiggly, and they noticed that, they said that, well, the only way this could even happen, because if it was a single-stranded, if DNA was single-stranded, it would just look like this. It would be straight lines, but they said that the only way this image of DNA could be even like, of this image could be about, is that if the DNA structure was double-stranded. So that is what they concluded. That was their thing. That was what Franklin said, that this is, it's DNA is double-stranded. And this was a big discovery at the time. And so she had held a conference with other scientists to kind of like show like her findings. And in that conference, there was a man who snuck into the conference, and his name was Crick. He had snuck into the conference and stole this information because him and his partner Watson, they were the ones who created the first 3D model of DNA. So, actually it was Watson who snuck into the conference. So they had made the whole 3D structure, but then they were just stuck on one thing. What is it single-stranded? Is it double-stranded? But then when Watson had snuck into the meeting and he found out that it was double-stranded, that was the last information they needed. So they created the first ever 3D structure of DNA. Was made out of like metal and plastic and things like that. And they're very worldwide known for this big discovery because, yeah. So those are all the, and